The topic of today's lecture is the synapse. A synapse is a connection between a neuron and another neuron. In the CNS, typically the second cell will be a neuron. In the peripheral nervous system, or PNS, the second cell will be a muscle or gland often. Occasionally it will be another neuron if it's, say, in an autonomic ganglion, but in the PNS, you're often talking about the second cell being a muscle or gland. When the connection is made with a muscle, it has a special name. It's called a neuromuscular junction. When you have two neurons that are connected to each other, the first neuron is called the presynaptic neuron. This is the one that sends the signal to another neuron. The second neuron is called the postsynaptic neuron, or receiving cell. You can see here between these two cells, there's a synapse. So here's the neuron, its cell body, it has dendrites, it has an axon, it has axon terminals. These are also called synaptic boutons. Bouton means button in French. And it makes a connection with the dendrites of another neuron. So this connection has a special name. It's called a synapse. Typically, you'll see in diagrams that the presynaptic cell can send a signal to a dendrite, but it's also possible for the presynaptic cell to make a synapse onto a cell body, and potentially it could also make a synapse onto an axon of second neuron. So the typical picture you'd see in a textbook would be an axodendritic synapse, meaning the axon terminals of the presynaptic neuron make a connection with the dendrites of the postsynaptic neuron. But of course, also quite common are axosomatic synapses. These are synapses from a presynaptic cell or neuron onto the soma or cell body of the postsynaptic cell. And finally, axoaxonic synapses. Axoaxonic synapses also exist, where an axon terminal makes a connection with the axon of another neuron. These connections tend to be very close to the terminal of the second neuron. In other words, they'd be directly on the synaptic bouton of the second neuron, and it'll actually regulate things like neurotransmitter release at that second neuron's axon terminals. So again, most synapses are in fact axodendritic, the typical picture you'd see in a textbook, and mostly synapses will send a signal in one direction. We can get, in a more advanced class, we might talk about retrograde signals that pass from the postsynaptic cell back to the presynaptic cell. But for our purposes here in this class, we'll mostly consider signaling in one direction. And finally, synapses can be both electrical or chemical. So in your body, you have quite a few electrical synapses. For example, you have electrical synapses between smooth muscle cells, say in the GI tract and in your stomach, and small intestine and so forth. Also, you have electrical synapses between cardiac muscle in your heart. In your brain, there are some neurons that make electrical connections with one another. And finally, you also have electrical connections between glial cells in your brain. These cells are joined by what are called gap junctions. So an electrical synapse consists of a number of gap junctions, and these gap junctions consist of protein that spans the gap between two neurons. Here's a figure from your book. This is these are cardiomyocytes, and what it's showing you is at the level of an intercalated disc, which is supposed to be shown here. It's a little hard to see, but this Y-shaped, inverted Y-shaped cell here is one cardiomyocyte, and here's another cardiomyocyte. So at the level of this intercalated disc, you have special protein that serve a mechanical function of keeping these two cells essentially joined together. But also at the intercalated disc, you also have these connexins, which form these gap junctions. So you can see these are proteins. So there'll be a number of proteins in one cell, another proteins in another cell, and they'll line up, and they'll form an aqueous pore that allows for the movement of ions, such as sodium, potassium, chloride, and calcium, from one cell to the next. And it'll actually allow for bigger molecules, such as ATP, glucose, and so forth, often can also pass through. Now, the actual, so this would be a typical vertebrate electrical connection, and there are different sizes of electrical connections, and that can be tested by putting, for example, dye with different molecular weights, and you could see you get a rough idea of how big these connections actually are. So chemical synapses are also present in your brain and within other parts of your nervous system, such as the peripheral nervous system. So synapses involve the release of a chemical called a neurotransmitter from the axon's terminal bouton. The synaptic cleft is the space between this terminal bouton or axon terminal and the next neuron, which is the postsynaptic cell. It could be a muscle cell here. So this synaptic cleft is very, very small, and basically it's held together because there are cell adhesion molecules, or CAMs, that essentially stick out of both the postsynaptic cell and the presynaptic cell, and they join together. So they essentially hold these cells in very, very close proximity one to another. This is an electron micrograph of an actual chemical synapse. You can see in the presynaptic cell there are a number of organelles such as mitochondria. So here's the terminal bouton of an axon, or axon terminal. And you can see all these small circles. So these are synaptic vesicles. So they're spherical bits of membrane, and within each of these vesicles are thousands of molecules of neurotransmitter. In this case, they're filled with acetylcholine, or ECH. And you can see actually here that there's a lot of reserve synaptic vesicles, and then there are some synaptic vesicles that are right at the level of very close to the membrane. So these vesicles are said to be docked. They're essentially ready to be released if an action potential were to invade this axon terminal. Then you can see the synaptic cleft. You can see it's very, very thin. So once the neurotransmitter is released into the synaptic cleft, it only has a very short distance to diffuse, which is good. And then here's the postsynaptic cell. And often on the postsynaptic cell, there's a lot of protein involved, such as receptors and so forth. So often you see this will be a little bit darker in electron micrograph. And then you can see that this is actually skeletal muscle, because you can see these very nicely arranged thin and thick filaments, these actin and myosin filaments. Okay, in the previous lecture, we talked about how action potentials can conduct along an axon. Well, eventually they'll conduct all the way to the axon terminal, which is shown here. So the action potential invades the axon terminal, so there's a change in membrane potential. It goes from resting membrane potential to about positive 30 millivolts or so. Now that signals voltage-gated calcium channels to open. So these respond to depolarization by opening. And if you recall, we talked about calcium previously in this class. So there's a very low concentration of calcium in the cytosol. There's a relatively high, well, it's lower than, say, other ions, but there's, in comparison to the intracellular calcium concentration, there's a high concentration of calcium outside. The difference actually in concentration is extremely, it's pretty extreme. It's about a factor of 10,000. So the concentration of calcium is 10,000-fold higher outside compared to inside. Calcium will move down its concentration gradient, and it's actually going down its electrical gradient also, because remember, the typical cell would be resting about negative 70 millivolts. So there's an influx of calcium. Calcium then interacts with a protein called synaptotagmin. This then interacts with vesicles, which are ready to fuse with the membrane. And so you can see what happens is the vesicular membrane can fuse with the plasma membrane. And when it does that, the neurotransmitters are released. And the protein that's involved in this docking and release is called, it's actually 3 different proteins. Together, they're called the snare complex. So they interact with the synaptotagmin complex, leading to this exocytosis. This slide is just a recap of what I just talked about. So in order for a transmitter to release, you have interaction between an influx of calcium and you have these synaptic vesicles that are docked and ready to be released. When calcium enters the cell, it binds to a protein called synaptotagmin, and that serves as a calcium sensor. The vesicles containing neurotransmitter are docked in the plasma membrane. They're held there by 3 snare proteins, which are collectively called the snare complex. The calcium synaptotagmin complex interacts with the snare complex, and the vesicle membrane fuses with the plasma membrane. This forms a pore, and then the neurotransmitter is released. So coming back to this idea, I just wanted to point out that as the synaptic vesicles fuse with the plasma membrane, they actually increase the amount of membrane in the synaptic bouton or axon terminal. And so necessarily there has to be some way to pinch off the membrane and form new vesicles. So that's not shown in this picture, but of course that is happening. So in the background, you have other processes that will pinch off membrane, form new vesicles, then the vesicles will then get refilled with neurotransmitter. Okay, this next slide is concerned with what happens to the neurotransmitter. So the neurotransmitter diffuses across the synapse, specifically the synaptic cleft, and there it will bind with specific receptor proteins. And there's some terminology we should know, which is neurotransmitter is often referred to as a ligand. And so this ligand is a general term. Ligand could also refer to something like a hormone. It's just a term for something that's a small molecule that attaches to a larger molecule, typically a protein. So these neurotransmitters will diffuse across the synaptic cleft. They'll then bind to these postsynaptic receptors, which are proteins. And so what happens is you'll have these receptors in the postsynaptic membrane, and these are often referred to as ligand-gated ion channels. So one class of receptor is the ligand-gated ion channel. When the receptor diffuses across the synaptic cleft and binds to this ligand-gated ion channel, the channel activates or opens, and then you'll have the flux of ions such as sodium into the cell. And that could then cause a depolarization in the postsynaptic cell. Okay, in the past lecture, we talked about action potentials. And I think we did touch a little bit on the idea that of a graded potential, but we'll go into a little more detail into that idea before. Remember that, let's say we have an axon. We'll draw up an axon here. If we were to inject current with our amplifier into the axon, so remember a triangle is our symbol for an amplifier. So we're going to inject, say, positive ions into this axon. Well, if we were to plot voltage versus distance, or D, right here where we inject the current, we see the greatest change in voltage. And as we move along the axon, say if we're recording at different spots along this axon, then we would see a decrement or an attenuation of the signal, and the same in the other direction. Okay, so that is referred to as a graded potential. It's something that that decrements over distance, and also depending on the amount of current we inject, we'll get different levels of depolarization. So say we inject less current, then we might get a slightly different curve. Now, it turns out this is just a sort of an artificial experimental demonstration of what a graded potential looks like. But within a cell, you can also have graded potentials. So for example, we can go back to showing voltage versus time. So let's say we have our cell that is a resting membrane potential of negative 70 millivolts. We then stimulate a presynaptic neuron. It releases neurotransmitter onto our postsynaptic cell. The neurotransmitter diffuses across the synaptic cleft. It binds to ligand-gated ion channels. Let's say there's an influx of sodium into the cell. Well, that will cause a depolarization. Now, eventually that ends and it goes back down to the resting membrane potential. So this would also be considered a graded potential. If we stimulated that same presynaptic cell a little more strongly, maybe it'll send more action potentials, or maybe we'll stimulate 2 presynaptic cells. There's various ways in which we could then stimulate a little more strongly and get a slightly bigger graded potential, okay? So the point is that we can get there's gradations in the amplitude when you have these graded potentials. And also the other important thing that I just pointed out is with distance, these graded potentials attenuate or become smaller. Now, this is different than an action potential. Recall that with an action potential, the action potentials don't attenuate. They keep, they're a regenerative phenomena. They'll just move along the axon and keep the same amplitude. Now, there are different types of graded potentials. You can have sodium fluxing in, or you could have calcium fluxing in. Those could cause graded depolarizations. And those are generally going to cause a depolarization, and that's going to be have an excitatory effect on the cell. So those are referred to as excitatory postsynaptic potentials, or EPSPs. If you open a potassium channel or a chloride channel, because of their equilibrium potentials, which are fairly negative, you'll often get a graded hyperpolarization. And that's sometimes referred to as an inhibitory postsynaptic potential, or IPSP. So I want to spend a little bit of time on defining these things a little bit better. The actual definition of an EPSP is it should be something that increases the probability of an action potential being generated. So let's think about what that means. So we have our neuron. Here's voltage and time in this graph. And sorry for the terrible drawing. So we'll have a depolarization, right? Now, that depolarization may not reach spike threshold. It could be below the, if our cell is at negative 70 millivolts, let's say we need to get to negative 55 millivolts to actually generate a spike. So this would seem to be sub-threshold. But we would still consider this to be potentially excitatory, because the way neurons work in reality, they're nothing like what we show in these pictures. They actually have essentially a barrage of input all the time. There's lots of synaptic input coming into them. So as long as the potential is moving it towards the towards the spike threshold, then we would consider it to be excitatory. Okay, so and again, you might have multiple events, and they might add up together. And if you have enough of them, you might actually get to spike threshold, right? And then you get the initiation of a spike. So those would be considered excitatory postsynaptic potentials because they increase the probability of an action potential being generated. Okay, so we'll move on next to IPSP. Okay, so the definition of an IPSP is very similar, just in the opposite direction. We have here voltage and time. So an IPSP will decrease the probability of an action potential being generated. So we have our neuron here with a resting membrane potential of negative 70 millivolts. Now, if we stimulate a neuron and it results in the opening of, say, potassium channels, and potassium fluxes out of the cell, then the cell will hyperpolarize. So that's very clearly inhibitory, because you're bringing the membrane potential further away from the spike threshold, and therefore you're making it less likely that the neuron would generate an action potential or spike. So IPSPs could be thought to as something that counters EPSPs. So you could have potentially EPSPs and IPSPs occurring at the same time. And these are going to, remember, the spike is being generated at the initial segment of the axon. So that's where all the integration and summation goes on. So if you had, imagine you have an EPSP or enough EPSPs that you would get to spike threshold, but simultaneously you might have an IPSP. Well, that might actually prevent the neuron from actually generating a spike. Okay, now, as your book points out, IPSPs usually move the membrane potential farther away from threshold. So that, in other words, they're often hyperpolarize the membrane, and that's exactly what I've shown here in this picture. I just want to, this is something for a more advanced class, but it's actually possible to have an IPSP that doesn't change the membrane potential at all, and yet it's still inhibitory. And that would occur if the equilibrium potential of the synapse, the inhibitory synapse, is essentially equal to that of the resting membrane potential. So it's not entirely true that IPSPs always hyperpolarize the membrane. They can either hyperpolarize the membrane, or they can bring the membrane to a potential that's below the spike threshold, and both of those would be considered to be inhibitory. So I just wanted to say that. Your textbook doesn't really go into the details of that because it's a little more advanced than this class we need to get for this class. But I just want to at least state that so we have the truth out there. So I want to recap some of the things we talked about because I know there's a lot we just covered just now. So action potentials are conducted by the axon. They arrive at the axon terminal, and there they lead to the opening of voltage-gated calcium channels. Those voltage-gated calcium channels open. Calcium enters into the presynaptic terminal. Calcium is essentially a signal that causes the presynaptic terminals that are fused with a membrane to release the neurotransmitter into the synaptic cleft. And also the synaptic vesicles that are sort of held in reserve, they'll actually move also towards a docking point. Once we have a neurotransmitter in the synaptic cleft, they diffuse very rapidly because you saw that there was a very short distance between the presynaptic and postsynaptic cell. Now, one mode of action for the neurotransmitters that may bind to a postsynaptic receptor. These may be ligand-gated channels. So these are channels that open in response to having a ligand bind to them, which would be the neurotransmitter. Then you might have the diffusion, if these were sodium channels, you might have the diffusion of, say, sodium into the cell. An influx of positive ions into a cell would cause depolarization. And a depolarization would be considered an EPSP, or excitatory postsynaptic potential. So for example, here in this picture, we see two synaptic boutons. They're releasing neurotransmitter, and there's a depolarization, which is shown in red here in the postsynaptic cell. Now, if this depolarization is great enough, it will attenuate as it moves through the cells, say the cell body, to the initial segment where the spikes are actually generated. But if you reach spike threshold in the initial segment, then you'll generate, postsynaptic cell will generate an action potential, and then that will move down the axon. Okay. So we have essentially a transition from these inputs occurring. You have a graded potential that occurs in the postsynaptic cell. If that graded potential is big enough, it reaches spike threshold, and then you have this all or none event, this action potential that always has the same amplitude and that can travel or propagate down the axon. Okay, so that's kind of a summary, a simpler summary of what we've just been talking. And this is just a picture from your book, an illustration showing you a little bit more graphically what would be going on. So you have, again, a presynaptic cell. It has axons that bifurcate. These would be considered collaterals, these little shorter bits of axon. And then they have the synaptic bouton or axon terminals. Here we're showing axonal somatic synapses. They would release transmitter. You'd have some sort of graded potential change in this postsynaptic cell. Let's imagine it's excitatory. Those potentials would decrement with distance as they move through the soma. But if they get to the initial segment and they're still above this combined synaptic potential is higher than the spike threshold, then you'll have the initiation of an action potential. In this case, we have a myelinated axon, so the action potential will jump. Remember, we have saltatory conduction from node to node, and these nodes are called the nodes of Ranvier. Finally, you'll get to the very end where you see these bifurcations and you have axon terminals, and the neurotransmitter is released onto the next cell, which could be another neuron, it could be a gland, it could be smooth muscle, it could be skeletal muscle. So your brain and your body use many different neurotransmitters. One of the most important is acetylcholine. So we're going to spend some time talking about it. This actually happens to be one of the first ones that was discovered. So it's interesting from a historical perspective. And it's actually important from an evolutionary perspective. Acetylcholine was actually a signaling molecule probably long before there were even nervous systems. It was already an important signaling molecule. So it was one of the first neurotransmitters used by animals. So as I mentioned, acetylcholine is really interesting in the sense it's such an ancient molecule. And for that reason, it's sort of ubiquitous, meaning it's everywhere within your body. So it's used as a neurotransmitter, and it binds to two different types of receptors. So ACH stands for acetylcholine, R for receptor, so ACHRs. In some cases, acetylcholine will have an excitatory effect. In other cases, it'll have an inhibitory effect, and this really depends on the organ involved. Let's take one example, which is the somatic motor neurons. These are neurons that innervate, meaning they will make synaptic connections or neuromuscular junctions with skeletal muscle. So somatic motor neurons release acetylcholine, and they always excite skeletal muscle. So we can see here within your spinal cord, you have, we've talked a little bit about gray and white matter. So this is gray matter. You see this butterfly-like shape here? This is all gray matter. So you have a lot of cell bodies and not a lot of myelin. In the white matter, you have a lot of tracks that are moving longitudinally. So that's why you have the whitish color to these other bits of the spinal cord. So this is a motor neuron, and it's within what's called the ventral horn here of the spinal cord. Now it sends an axon out. This is a spinal nerve. You can see it's myelinated. You see this Schwann cells, so it's doing myelination. You get to the end, and you have bifurcations, and then you have the axon terminals or synaptic bouton onto this postsynaptic cell, which happens to be a skeletal muscle. And so acetylcholine is released, and then there are receptors here on the postsynaptic cell that will be ligand-gated ion channels. So they'll open in response to the release of acetylcholine, and this will lead to excitation of this postsynaptic cell, the skeletal muscle. Now these are referred to as nicotinic acetylcholine receptors. And the reason for that is scientists have done experiments, and they showed that these particular receptors can be stimulated by acetylcholine itself, which is the natural endogenous neurotransmitter in your body. They can also be stimulated, however, by nicotine. So nicotine, of course, comes from things like the tobacco plant, and so it can also stimulate, so it essentially acts as a toxin, and it can stimulate skeletal muscle also that through this exogenous compound, one that comes from outside of your body. So nicotine is just one type of receptor, so we'll talk about another type here. So the autonomic motor neurons are neurons that release acetylcholine, and they can have potentially an excitatory effect. So for example, here's a autonomic motor neuron. So recall that the autonomic system is a system that, as the name implies, is sort of automatic. It does a lot of things, like it is involved in regulating the gastrointestinal tract. It's involved in sweating, so it innervates glands. It also innervates smooth muscle, such as the muscle of the uterus and things like that. Okay, so here's an autonomic motor neuron. It's sending an axon out into the peripheral nervous system. It terminates here in what's called an autonomic ganglia. So you can see the synaptic bouton is making a connection with the postsynaptic cell, which is another autonomic motor neuron. Now at this connection, the postsynaptic cell has nicotinic receptors, and this effect is excitatory. In other words, acetylcholine is released, goes across the synaptic cleft, binds to these nicotinic receptors, which are ligand-gated receptors, which are also ion channels, and they'll allow the influx of, say, sodium into these postsynaptic cells. The postsynaptic cell will depolarize, it'll generate an action potential, the signal will continue all the way to the effectors. So here we're showing a variety of effectors. So autonomic motor neurons can go to lots of different effectors. So on the target or effector, they'll often have an inhibitory effect. So they'll innervate, for example, smooth muscles, cardiac muscle, certain glands. And often these postsynaptic targets here will have muscarinic acetylcholine receptors. So this is a very different type of receptor. And the effect is going to be inhibitory rather than excitatory. So this is just an illustration, really, of the fact that You have one neurotransmitter, and yet you can have opposite effects. You can have an excitation if the postsynaptic receptors are nicotinic receptors, and you can have inhibition if the postsynaptic receptors are muscarinic. So let's recap a little bit of what we talked about. There are two types of acetylcholine receptors. There are nicotinic acetylcholine receptors. These can be stimulated by nicotine, which of course isn't natural. It's not found in your body. It's found in the tobacco plant. Nicotinic receptors are found on skeletal muscles. They're found in autonomic ganglia, as I just showed in the previous slide. They're also found in some parts of the central nervous system. So within your brain, you have lots of nicotinic ACH receptors. In fact, that's why nicotine is an effective stimulant, because it's acting on the nicotinic receptors within the central nervous system. You also have muscarinic ACH receptors. These can be stimulated by muscarine. So again, this is an exogenous compound coming from outside of the body, and it comes from poisonous mushrooms. Muscarinic ACH receptors are found in the CNS. They're found on smooth muscle, they're found in cardiac muscle, and they're found in certain glands that are innervated by the autonomic motor neurons. When you're talking about neuroscience, there's a lot of terminology, and one bit of terminology that's very important is the term agonist. Agonist is a drug that can stimulate a receptor. So we just talked about nicotine and how it can stimulate nicotinic ACH receptors. So here, these of course come from the tobacco plant. So the nicotine is an agonist. It binds to nicotinic ACH receptors. It opens them or activates them, and it has a very similar effect to that of the regular endogenous neurotransmitter, which is acetylcholine. So it would be considered an agonist. Muscarine from poisonous mushrooms, it will bind to muscarinic ACH receptors, and again, it'll have an activating effect. It'll open it won't open these channels, but it'll stimulate these channels. We'll talk more about how they work in a second. It'll stimulate these channels, and so because it has a similar effect to the actual natural endogenous neurotransmitter, which is acetylcholine, it would also be considered an agonist. Okay. Another very important term is antagonist. An antagonist is the opposite of an agonist. It's a drug that will bind to a receptor, but it will inhibit the receptor. It'll have the opposite effect. So in other words, for a good example would be atropine. This is an antagonist for muscarinic receptors. So when atropine binds to a muscarinic receptor, it prevents the muscarinic receptor from being activated. So even if you had acetylcholine present, it'll compete with the acetylcholine and prevent the activation or stimulation of the muscarinic receptors. So a very famous example of this would be, atropine comes from certain plants like belladonna. So belladonna literally means beautiful woman in Italian. And the reason for this was that people knew that they took extracts from the berries of this plant, and if you put that in your eye, it'll actually make the iris relax, and so you have a dilated pupil. And so that was considered beautiful in the past, and therefore the term belladonna. So that was a very early use of atropine to block muscarinic receptors and therefore relax this muscle, and so that the eye would appear, I guess, more seductive. So another example of an antagonist would be curare. This is an antagonist for nicotinic receptors, and so this prevents. It'll bind to nicotinic receptors, but it'll prevent nicotinic receptors from being activated. Recall that many of the nicotinic receptors are, as I mentioned earlier, they're ion channels, and they allow for the flux of sodium into a cell, and therefore the depolarization of the postsynaptic cell. So, for example, at the neuromuscular junction, you have nicotinic receptors, as I mentioned earlier. So normally when those are stimulated by acetylcholine, you get depolarization of the postsynaptic cell, which is the muscle, and you get then muscle contraction. So curare will bind to the nicotinic receptors, but prevent them from opening. So this means that the skeletal muscle will not be able to generate an action potential. It won't be able to depolarize. And therefore, the muscle is paralyzed. It won't be able to contract. And you have what's called flaccid paralysis, meaning that the muscles are just flaccid. They're not able to contract. And so this was, these, curare would come from trees in the Amazon rainforest. And so hunters would make curare darts, and they would use a blowgun to then essentially poison animals that they were hunting. The animals would be paralyzed, and then they could capture the animal and, obviously eat it. Okay, previously we talked about the idea that a neurotransmitter can bind to a receptor, and that receptor is itself an ion channel. And you can have that, and this can then lead to the influx of sodium, which would then say depolarize the cell, and potentially the postsynaptic cell might then generate an action potential. So those are referred to as ligand-gated ion channels, or we could just say ligand-gated channel. But there's another way that receptors can open a channel, and this route is referred to as G protein coupled channels, or sometimes are referred to as G protein coupled receptors. Let's first talk a little bit in more detail about these ligand-gated channels. So the receptor protein is also an ion channel, as I mentioned earlier. So the neurotransmitter binds directly, and this leads then to the opening of the ion channel. Nicotinic ACH receptors are ligand-gated channels with 2 receptor sites for 2 acetylcholines. In other words, it'll only open when 2 acetylcholine molecules binds to it. And then this particular channel actually allows for both sodium and potassium to pass through it. And in fact, the permeability of this channel is equal for sodium and potassium. There's really no difference. So let's think about what would happen. So you have the 2 acetylcholine molecules binding to this nicotinic acetylcholine receptors. Recall that there's more sodium outside compared to inside of a cell. This inside of the cell is also negative with respect to outside when the cell is at rest, at resting membrane potential. So therefore, sodium will flow down its both electrical and chemical gradients. Now, at the same time, potassium will flow out. There's a lot more potassium inside, so it's flowing down its chemical gradient. The inside of the cell at rest is negative with respect to the outside, so it's actually flowing against its electrical gradient. But remember, we talked about the Nernst equation. So with the Nernst equation, we essentially figure out an equilibrium potential. So generally, as long as the cell is above, the membrane potential of the cell is above the equilibrium potential for potassium, we know that potassium will always be flowing out. Now, it turns out, and again, we're not going to go into a great level of detail in this particular class on this, but what's referred to as the driving force for sodium is greater than that driving force for the potassium. So, and I sort of alluded to why this is. We have both, for sodium, we have both an electrical and chemical gradient that are in the same direction that would drive sodium in, whereas for potassium, you have a chemical gradient which would drive potassium out, but the electrical gradient would actually tend to retain potassium in. So the driving force for potassium movement is less than that of the driving force for sodium. Therefore, you would expect, even though this channel is equally permeable to both ions, there'll be more movement of sodium in. There'll be a higher net flux of sodium into the cell than there would be a net flux of potassium out through these particular channels. Therefore, the overall effect would be to depolarize the membrane. So you would have an EPSP, or excitatory postsynaptic potential, in response to opening of these receptors. This is an illustration from your book showing exactly what would happen. So in this first case, you can see here's a nicotinic receptor. It's closed, so this gate is closed. Acetylcholine, or it could be nicotine, binds to the receptor, but if only one site is occupied, the channel is still closed. Once both sites are occupied, in this case by acetylcholine, then the channel opens. Sodium will move in, so there'll be a net movement of sodium into the cell, which would depolarize the cell. There's also a net movement of potassium out of the cell, which would tend to hyperpolarize the cell, because you have positive charges leaving. But as I just discussed, the overall net number of ions is such that there will be a net increase in positive ions inside of the cell. Therefore, this activation of this receptor will lead to a depolarization of the postsynaptic cell. As I mentioned earlier, there may be multiple synapses from a presynaptic cell onto a postsynaptic cell. Also, you may have multiple neurons converging on the same postsynaptic cell. Therefore, if we take a single stimulation of a single synaptic bouton, we can imagine there'd be inflow of sodium, which would depolarize the cell, and that would create an EPSB, or excitatory postsynaptic potential. These can occur in the dendrites as well as the cell bodies. So that's the case for, say, just a single synaptic bouton being activated. But let's imagine there's multiple EPSBs all coming together at around the same time. These could add together and create a greater depolarization. So in other words, they would summate. And remember, the EPSBs are graded potential. So unlike an action potential, which are all in none, these can add together. So then you have the summation of multiple EPSBs. And this may cause the initial segment of in the axon to reach spike threshold, and that could then lead to the generation of an action potential. This is an illustration from your book showing exactly this concept. So we have a cell that's at rest. It's at negative 70 millivolts. Perhaps we might have one of these synaptic boutons being stimulated, so you get a certain EPSB of a certain size. But now imagine you have two being activated at the same time. You might get a greater a summation of these two EPSBs and a greater change in membrane potential. Let's imagine now that we have four of these synaptic bouton activated at the same time. This would lead to an even greater depolarization, or greater amplitude of EPSB. In this case, we're reaching spike threshold, for this, which in this neuron is negative 50 millivolts. This leads then to an all or none event, which is the action potential occurring. Okay, I realize it can be quite confusing to think about action potentials versus these graded potentials, such as excitatory postsynaptic potentials. So this is a table from your book, and we'll go through some of the characteristics of them, these two types of potentials. So if we just look at action potentials, they have an amplitude that's fairly consistent. They're always going to go to about positive 30 millivolts. So these are considered to be all or none events. What stimulates them to occur? Well, often you could have the opening of some ionic gates, allowing for sodium to flux into the cell. This could cause depolarization. And as long as you get to the spike threshold level, in other words, if the stimulus is supra-threshold, then you'll get a full-blown action potential occurring. The initial effect of the stimulus is that sodium channels will open. And remember, we talked about the fact that as sodium channels open, they'll lead to sodium influx. The sodium influx will depolarize the membrane of the cell, leading to even greater activation or opening of sodium channels, which leads to more influx of sodium, and so forth. Now, eventually, the action potential has to come down, right? It can't simply stay up there for a long period of time. That would damage the cell. In fact, it would kill it. And so the membrane has to repolarize. So the falling phase of an action potential is due to the opening of voltage-gated potassium channels. Now, those are also open in response to depolarization, just as voltage-gated sodium channels do. But they're slower to open, and so they open with a delay. So that allows the membrane potential to escape upwards, and then as they open, they then bring the membrane potential down. And aiding in the falling phase of the action potential is the inactivation of sodium channels. I won't go through all that again, but I did talk about it in detail previously. So conduction distance. So action potentials are regenerative, meaning they keep propagating or over the length of an axon. So they'll move and have very similar shape, amplitude, and width, and so forth as they move along an axon. Positive feedback, yes. So an action potential during the rising phase relies on positive feedback between depolarization and opening of sodium gates. The maximum depolarization, as I said, is around, I've been saying plus 30, your book says plus 40 here, but it's somewhere in that range of about plus 30 to plus 40 millivolts. Action potentials do not summate. There's no, they're all in non-events. Action potentials have a refractory period. There's a period immediately after an action potential where you have the absolute refractory period in which you can't generate another spike. A little bit after that, you have a relative refractory period in which it's possible to generate another action potential as long as you inject enough current into the cell or there's enough depolarization of the cell's membrane. So action potentials are affected by drugs. So one of the most famous drugs that affects action potential is tetrodotoxin. Tetrodotoxin comes from the pufferfish as well as some other fish. It blocks voltage-gated sodium channels. So this pufferfish is considered to be a delicacy in Japan. So people do eat the flesh of the pufferfish, but you have to be very careful from preparing this dish. And people actually do die every year from poisoning. Essentially, if you block your voltage-gated sodium channels, you can imagine what would happen. You wouldn't be able to generate action potentials. And so motor neurons going to, say, respiratory muscles, such as your diaphragm, would not be able to generate action potentials, and so you wouldn't be able to breathe. And so it's probably a pretty horrible death. Okay, I'd like to go through a quick rundown now of excitatory postsynaptic potentials. We'll compare them to what we just discussed for action potentials. So action potentials were all or none. Excitatory postsynaptic potentials are graded, meaning that they can have different amplitudes, and also they'll attenuate over distance, as we discussed earlier. Action potentials are open in response to depolarization. EPSPs occur because acetylcholine or other neurotransmitter has been released, and this acts on postsynaptic receptors, potentially causing ion channels to open, and therefore the influx of sodium or other ions. The initial effect of the stimulus is that it can cause, in the case of an action potential, sodium channels open. For an excitatory postsynaptic potential, you have these receptors which actually allow both sodium and potassium to move through them. But as I discussed earlier, sodium influx will be greater than this potassium efflux, therefore you have a depolarization. Now, cause of repolarization. So EPSPs, they end eventually. And why does that occur? Well, one reason is we've sort of alluded to, which is the positive charges that enter into the cell will actually dissipate as they move away with distance from the initial site. And actually, a lot of we do have leak channels in our membranes, and so these charges such as sodium and potassium, things like that, would actually be able to leak back across the membrane. Conduction distance. So, as I mentioned earlier, excitatory postsynaptic potentials are graded, and so you'll have a localized large change in memory potential, but with distance, it'll decrease. There's no positive feedback with EPSPs. Now, the maximum depolarization for an action potential is around plus 30 to plus 40 millivolts. For an excitatory postsynaptic potential, the maximum depolarization is about 0 millivolts. And it's rare that you'd ever actually reach 0 millivolts. But this is, in fact, the equilibrium potential for the excitatory postsynaptic potential, which is something we can calculate, but it's a little bit beyond the scope of this class, so we're not going to do that. But essentially, it can bring the membrane potential to a maximum of about 0 millivolts. Summation, so EPSPs can summate, producing graded depolarizations of greater and greater size. And of course, action potentials don't summate. Excitatory postsynaptic potentials do not have a refractory period. So they're different than action potential in that respect. And effects of drugs, ACH is inhibited by curare, but not by tetrodotoxin. So curare, remember, is the poison in these blow darts used in South America to paralyze prey. And the way it works is it blocks the nicotinic acetylcholine receptors. And therefore, in doing so, it blocks the ability of the postsynaptic cell, such as skeletal muscle, to depolarize and generate an action potential. And that's necessary for the skeletal muscle to contract. So curare causes what's called a flaccid paralysis, meaning that the muscles are unable to contract. And that's how the hunter then is able to then pick up the animal and, cook it and eat it. So far, we've been talking about ligand-gated channels that are also ion channels, which are a very common type of channel. But equally common are G-protein coupled channels, also called G-protein coupled receptors. And these work in a slightly different fashion. The neurotransmitter receptor is actually going to be a separate protein than the ion channel. And so what's going to happen is that the ligand will bind at the receptor, and then you will have opening of the ion channel, but it's going to occur in an indirect fashion through the use of a G protein. And I'll show a picture of this in a second. In this indirect manner, muscarinic ACH receptors may cause ion channels, such as potassium channels, to either open or close. So this is a very nice illustration of muscarinic receptors and how important they are. So muscarinic ACH receptors require G proteins. The heart is an organ that generates its own rhythm. In other words, it's referred to as myogenic. And the reason it generates its own rhythm is because you have in the upper right atrium something called the SA node, or the sinoatrial node. Within that node, you have special heart muscle cells that have been modified. They don't participate in contraction. They participate simply in generating a timing signal for the rest of the heart. So these are referred to as pacemaker cells, or pacemaker cardiomyocytes. So it turns out that, well, we talked a little bit about the autonomic nervous system. Well, you have nerves that go directly to the heart, so they directly innervate the heart, and they release acetylcholine. And this acetylcholine will have an effect on the speed of the heart rate, on the heart rate. So in other words, how fast your heart beats. So here's a picture from your book. Acetylcholine is released at a synapse or a neuromuscular junction. It then binds to a receptor, which is a protein. Now, this protein is called a G protein coupled receptor because it's coupled here with a G protein. Now, G protein actually has 3 subunits, alpha, beta, and gamma. And in this particular case, what we're seeing is that the beta and gamma translocate together. In other cases, the alpha may translocate. So it depends a little bit on the receptor. In this case, the beta and gamma are translocating through the membrane until they reach a channel. This happens to be a potassium channel. So the G protein binds to the potassium channel, causing it to open. And this channel, it's a potassium channel. It doesn't allow sodium to pass through it. And so potassium will flux out of the cell, okay? So here's a question for you. Will the opening of the potassium channels cause the pacemaker cell in the SA node of the heart to depolarize or hyperpolarize? I'll give you a moment to think about that. Okay, so let's think about this. So we'll have positive charges leaving the cell, leaving behind negative charges. And so that should hyperpolarize the cell. And we haven't really discussed this yet, but you could also guess, will the heart rate increase or decrease? It turns out the heart rate will decrease because you're hyperpolarizing the cell. So this is essentially acting sort of as an IPSP, which we did discuss earlier. So it's bringing the cell further, its membrane potential down further, and it's going to delay the generation of an action potential. It's actually a little bit more complicated than what I'm saying right now, but it does sort of make intuitive sense that if you hyperpolarize the cell, it'll take longer for the cell to generate its next action potential, and so you'll slow the heart rate. Okay, to summarize what we just talked about, G protein coupled channels are very important. Binding of acetylcholine opens potassium channels in some tissues. In the heart, potassium channels are opened by the beta-gamma complex, creating IPSPs, or hyperpolarization, that slow the heart rate. Again, these channels are so important, it's worth really understanding them a little bit better. So the G proteins, as I said, have 3 subunits, the alpha, beta, and gamma. Binding of one acetylcholine molecule results in the dissociation of the alpha subunit. And either the alpha or the beta or gamma can then diffuse through the membrane. You can translocate laterally to an ion channel. This can then open a channel for a short period of time. The G protein subunits then dissociate from the channel, and then the channel closes. And we'll talk a little bit more of how this occurs in a second. Before we get into some of those details, let's talk about smooth muscle. So smooth muscle also has muscarinic acetylcholine receptors, or MACHRs. In smooth muscle cells of the stomach, binding of acetylcholine to muscarinic acetylcholine receptors causes alpha subunits to dissociate. And then these bind with gated potassium channels. In this case, you have a different situation, though. These potassium channels are open already. And what happens when the alpha subunit binds these potassium channels is that they'll close. So the question is then, will smooth muscle cells of the stomach depolarize or hyperpolarize when this all occurs? Think about that for a moment. Okay, so normally these potassium channels are open. So presumably you also have weak potassium channels. So remember, potassium tends to want to drive the membrane potential downwards, or hyperpolarize the membrane potential towards EK, which is generally about negative 90 millivolts in most cells. So if these are open most of the time, then these potassium channels, together with the leak channels, are driving the membrane potential downwards towards EK. Now, the leak channels will stay open, right? Those don't open or close. They're more or less just constantly open. But these channels, potassium channels, will close, and therefore they will exert less of this downward effect. And you have other leak channels, like sodium leak channels and chloride leak channels, and they'll tend to drive the membrane potential up a little bit. And so the balance is shifted now. You'll have less potassium permeability compared to the sodium and chloride permeabilities, and therefore the cells should depolarize. So just to restate what I've just said, so binding of acetylcholine may close potassium channels, and this will cause a depolarization, and that's going to be an EPSP, excitatory postsynaptic potential. So in smooth muscle of the stomach, potassium channels are closed by the alpha subunit, producing EPSPs, or depolarization, and this leads then to the contraction of these muscles.
As I mentioned earlier, G protein coupled receptors are among the most important class of receptors there are. And so they're the target of many therapeutics, many drugs, so forth. And so it's worth going a little more into detail how these actually work. So basically, the alpha receptor is bound to GDP when the alpha receptor, the beta, and the gamma all as a complex together bind to the receptor. Now, when the regulatory molecule, which could be a ligand like a neurotransmitter hormone, binds to the G protein coupled receptor, then you have a situation in which the alpha unit will essentially exchange GTP, guanosine triphosphate, for GDP, guanosine diphosphate. So now this alpha unit is activated, and at this point, this complex alpha, beta, and gamma comes apart, and the alpha may translocate laterally in the membrane to attach to an effector, and then have some sort of cause some sort of change or stimulation of the effector. And also the beta and gamma can also translocate, and they could potentially bind to another effector and also cause a change. Now, eventually, the alpha subunit will hydrolyze guanosine triphosphate to guanosine diphosphate. So we'll go back to alpha with the GDP attached, and you have inorganic phosphate released into the cytosol. And at this point, the alpha is able to move backwards and reassociate with the beta and the gamma. And then they can all, this complex can then reassociate with the original receptor. And on and on. And this could go around many times, of course. This is a table from your book, essentially describing the exact phenomena I just described in the previous slide. Okay, so one problem that can occur is once the acetylcholine is released into the synaptic cleft, it will then bind to the receptors. And this is not a covalent bond. There's a certain affinity between the receptor and the acetylcholine molecule, but it can unbind also. So it's a process of binding, unbinding, another molecule of ACH then will bind to the receptor and so forth. And this could essentially go on for a very long time until the ACH diffused away. And that would not be effective for a synapse. A synapse should be active for a short period of time, causing some sort of change in the postsynaptic cell, such as a depolarization. To prevent the continual stimulation of the receptors, in the postsynaptic membrane, there's an enzyme called acetylcholine esterase, or ACHE. And this hydrolyzes ACH or acetylcholine into acetate and choline. And both acetate and choline can be taken back into the presynaptic cell and then reused to form new acetylcholine. And so this is a way for the effect of the release of transmitter to be terminated. There are a number of drugs that are inhibitors of the action of acetylcholine esterase, ACHE. So these cholinesterase inhibitors essentially block the action of ACHE, and therefore they increase the amount of acetylcholine in the synaptic cleft after a synaptic event. In other words, the neurotransmitter is released into the synaptic cleft. Normally it's broken down or hydrolyzed by acetylcholine esterase. If you block the action of acetylcholine esterase, then the neurotransmitter is able to stay in a high concentration in the synaptic cleft for a longer period of time, and therefore continue to keep stimulating the postsynaptic cell. So this is a way of enhancing cholinergic synaptic transmission. So some cholinesterase inhibitors are used to treat autoimmune disease called myasthenia gravis. It's also important for the treatment of Alzheimer's disease. Cholinesterase inhibitors are also used, unfortunately, in World War II. These were used as nerve gases. So they would essentially overstimulate the synapses in the poor people that are exposed to this, and this could lead to paralysis and death. And in a very similar manner, you have organophosphate pesticides, and these kill insects by inhibiting ACHE. And again, it's going to overstimulate the cholinergic synapses. Again, it could lead to, when you overstimulate a synapse, for example, a neuromuscular junction, you can essentially lead to a kind of paralysis in which you're activating the muscle and contracting it over and over again. And so the insect or the person, in the case of nerve gas, are then paralyzed. Organophosphate pesticides such as chlorpyrifos, again, kill insects by inhibiting ACHE, leading to overstimulation of the ACHE receptors, acetylcholine receptors. Now, it turns out that chlorpyrifos has been shown to cause brain damage in children. There's very strong evidence for this. And so a number of states have started to ban it. So so California has a ban, I think, that will go in place. Hawaii already has a ban in place for this type of insecticide. Okay, let's go a little more in detail and talk about the neuromuscular junction. So somatic motor neurons, remember, they innervate skeletal muscle. And so they form what is called the neuromuscular junction with the muscle cell. So this is essentially a synapse. It's just in the periphery, and it has a little bit of a different structure, and so that's why it's given a different name. The area of the muscle cell with the receptors for neurotransmitter is called the motor end plate. So if we were to just look at the postsynaptic cell, this area where you have receptors, which I'm pointing to, would have a very high density of receptors, and it also shows this infolding. So it's a very special bit of membrane. That's called the motor end plate. And so what would happen is you'd have an action potential come down into the synaptic bouton. Calcium entry would occur. Neurotransmitter would be released into the synaptic cleft. It would bind to the receptors here. And at the neuromuscular junction, these receptors are ligand-gated channels, ion channels, that are permeable to both potassium and sodium. As we discussed earlier, there'd be more influx of sodium relative to the amount of efflux of potassium. Therefore, there'd be a depolarization. And that depolarization is called an excitatory postsynaptic potential. Now, there's a special name for these because it's a neuromuscular junction. You could call it EPSP, that's correct, but you can also call it an end plate potential, or EPP. The end plate potentials are due to the open voltage gated sodium channels. Remember, these are also permeable to potassium. The EPPs, which were through these ligand gated potassium / sodium channels, when they depolarize, they're going to activate, the depolarization itself will activate voltage gated sodium channels, just as in the initial segment of a neuron. And you'll get the resulting action potentials. There's, of course, voltage gated potassium channels and all that. And so the actual process of action potential generation is very similar to that which we discussed before, so I'm not going to go through it again. It's almost identical to that of in a neuron. Now, the action potential will be generated here, and actually skeletal muscle fibers are extremely long. And so the junction, these neuromuscular junctions generally are going to be towards the middle of the muscle fiber or muscle cell. And then the action potential will essentially go in both directions. And so they'll essentially cover the whole cell in that way. And then this electrical event, which is referred to as excitation, then leads to, with a slight delay, to the actual contraction of the muscle fiber. Okay, so let's talk a little bit about interruption of neuromuscular transmission. There are certain drugs that are very important in medicine. And what they can do is they can block neuromuscular transmission. Curare, we've already talked about. This is an antagonist for acetylcholine receptors. Remember, it binds to nicotinic acetylcholine receptors, which are the ones that you have in your CNS, but also in the PNS on skeletal muscle. It blocks those muscle ACH receptors. It does so by binding to them, but not allowing these ion channels to actually open. So the muscles are prevented from contracting. So essentially competes for the natural endogenous neurotransmitter, which is acetylcholine. And as we talked about, this was the poison that's used in the blow darts and the Amazon. So the poison originally comes from trees. And then it's put on the ends of these blow darts, and then you use it to paralyze and even kill the prey. And the way it kills animals is by paralyzing the diaphragm, which is the major muscle used for breathing. And so the animal will essentially suffocate. Curares actually can be used clinically as a muscle relaxant, of course, in much lower concentration. Another very important toxin is botulinum toxin. It's a neurotoxic protein that prevents the release of the neurotransmitter acetylcholine from axon endings at the neuromuscular junction. And it therefore also causes flaccid paralysis, just as curare does. Now, the mechanism is a little bit different. It's preventing the release of the neurotransmitter, okay? It is produced normally by the soil bacterium called Clostridium botulinum and related species, but it can be found inside cans of food that have been improperly processed. So, of course, normally the canning process is under very strict regulations and guidelines and so forth, and the machinery is all working. But occasionally, what will happen is they'll say overheat the cans, and there'll be a period of time when they seal the can where there'll actually be possible for bacteria to get in. And then those bacteria, this Clostridium botulinum, are actually perfectly happy to live in an anaerobic environment. So they'll multiply and they'll create toxins. And it's one of the most potent neurotoxins there is. So even a very small amount of it, you can't just, see, gee, I wonder if this is okay, and then taste it because you could be dead. And actually, people that do home canning have to be very aware of this. You have to do everything properly because, again, you could inadvertently grow this bacterium, Clostridium botulinum, and even just inhaling a bit of it or touching and so forth, it's so incredibly toxic that you could, end up in the hospital. Now, interestingly enough, small doses of this toxin can be used to paralyze or weaken muscles to cause facial wrinkles. And this goes by its then trade name called Botox, which again is just short for botulinum toxin. So here's a woman in the before picture. The doctors injected botulinum toxin into her forehead, perhaps other places in her face, and you can see that the muscles have relaxed, giving her a much smoother appearance on her forehead. And actually, movie directors have complained about this. They find that so many actresses and actors have used this that they are not able to emote. They can't show emotions on their face because their foreheads and other parts of their faces are essentially paralyzed. This is a table from your book, and I've actually blanked out a few of the toxins that we haven't talked about. We're going to restrict our discussion just to these 4 toxins. So you should know botulinum toxin. It's produced by Clostridium botulinum, a kind of bacteria in the soil. It inhibits the release of acetylcholine, or ACH. Curare, it's a resin from South American trees used in the blow darts. It prevents the interaction of, essentially is an antagonist. It's a competitive antagonist of acetylcholine. So it attaches to nicotinic receptor proteins on skeletal muscle, and therefore it blocks muscle contraction. So it leads to what's called flaccid paralysis. Tetrodotoxin is another very important toxin. It comes from pufferfish. It blocks voltage-gated sodium channels from opening, and therefore it blocks action potential. So again, that would lead to paralysis and death. And then we've talked about nerve gas a little bit. It inhibits acetylcholine esterase and postsynaptic membrane. And in doing so, it would essentially lead to overstimulation within the brain, but also at the neuromuscular junction. And again, this could lead to paralysis and death. And grouped together with nerve gas would be the organophosphate pesticides. Those work in exactly the same manner. They inhibit acetylcholine esterase in the postsynaptic membrane, and this leads to overstimulation of the postsynaptic cell and can lead to paralysis and death. And in much lower dosages than are used to kill insects, certain organophosphates, such as chlorpyrifos, can lead to brain damage in infants, and therefore the government's acting very slowly, but certain states have now banned it from use in agriculture.